All right, welcome back to part 4 of molecular structure of DNA and RNA. So if you recall, we had discussed the double helix. We had discussed how we have the backbones running on the outside, two strands running around each other. They are anti-parallel, so one is 5' to 3', to where the other one is 3' to 5'. And this helical structure wraps around with the base pairs, or the two nitrogenous bases that bond to each other. So these base pairs in the center stacked up on top of each other. Notice that these have a flat shape to it, and they stack directly on top of each other. This is very similar to paper stacking up. And the interesting thing about this is the ring structures that are present form van der Waals interactions between each other. So there's a mild adhesion between them all once they've gotten all wrapped up. And this is actually what is providing a lot of the stability of this double helix in and of itself. It's that and the combination of the restriction of various different bond angles. So, recognizing that the angles between two atoms can't just move around freely, they can rotate but not move. This leads to there being a minor groove, and if you recall the minor groove, a key characteristic is that you cannot access the nitrogenous bases inside. The major groove, you can access the nitrogenous bases. So something small enough could reach in and touch and touch. This means that if we have a protein that is binding DNA non-specifically, is probably going to be recognizing the minor groove and the backbone, to where things that are recognizing a specific sequence, such as TGCA, it is binding into the major groove. Now, all of this is in a type of DNA structure that we call bDNA. Okay? This is when we describe the double helix, this is what we're referring to. There are two other forms of DNA, alternative conformations that it can take. zDNA that the book talks about, and aDNA that the book does not talk about. I'm going to go ahead and mention those here. Now, these arise under specific conditions, so you can force it by altering salt concentration. Of course, that wouldn't really happen inside of a cell. But another thing that can force these conformations is if you have a protein that specifically binds the Z conformation, the interaction of the DNA in that protein can force it to take the Z conformation. And if those proteins link to each other, it may be able to cause large stretches to change conformation. All right, so here's the image from the book, and it gives you a side-by-side comparison of bDNA and zDNA. I'm just going to go ahead and progress on here so that we can see the A form with it as well. So you can see that both A form and B form are right-handed helices, right? So if you have it spiraling away from you, it is spiraling in a clockwise direction. zDNA, however, is actually in a left-handed. So it moving away from you, it will rotate counterclockwise. zDNA is ever so slightly thinner, but it is a bit more stretched out. aDNA has about the same width, but it is compressed a bit. And then we have our traditional bDNA form. What's significant about this is that Z-form DNA can be linked to gene expression patterns, and we'll go into more detail about this later. aDNA form is something that is also very common for RNA. Some of the different chemical structure of RNA, some of the fact that you have that oxygen in the backbone leads to a slight compression, and it tends to favor an A form of, in this case, a RNA. So RNA does tend to take this. DNA usually takes this, but it can be coaxed into either one of these under the proper conditions. Now, the book also talks about the fact that it was discovered that you can have a triple helix. While we don't understand the full significance of this, what we do know is that we can utilize this triple DNA structure to create probes to find out specific sequences. So how this works out is you essentially have another strand that is fitting into the major groove. And the base pairing rules are that a T can bind to an AT pair, and a C can bind to a GC pair. And what I want you to recognize here is that it's not binding in the same way that the two base pairs are binding each other. It's just coming in and making specific contact points. So by taking a specific sequence, we might be able to design a piece of DNA that we could send in and probe for something. There is some evidence that this might be involved in some gene regulation aspects, but again, we are still at the very beginning of understanding that. Now, the other thing that I want you to walk away with is some key structural features of RNA. And RNA is largely working the same as DNA, but we always think of it as a single strand. And unlike DNA, which can go on for millions of base pairs, RNA strands can be 100 to several thousand nucleotides in length, but they don't go into the millions of bases. Let's go ahead and take a look at the structure here to point out some key differences. So here we have the phosphodiester backbone. So we have phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Still has that same 5' to 3' directionality. So here's phosphate, carbon 5, carbon 3, phosphate, carbon 5, carbon 3, and so forth. The one major difference is this is ribose, not deoxyribose. So we have an OH group at carbon number 2 instead of just a hydrogen. This actually destabilizes the RNA and makes it more prone to degradation by high pH. The other thing that we see, which is a major difference, is we have replaced T with U. And again, this excuse me, largely serves as a marker for the fact that this is RNA, not DNA. All right. So as we were saying before, we usually think of RNA in terms of a single-stranded molecule, specifically thinking about messenger RNA, because that's where we see it over and over and over again. But even in messenger RNA, there are regions that are double-stranded. And these usually come to be by a single strand of the DNA having a complementary inverted sequence somewhere later down that can bind to itself. This allows for short regions to form a double helix. As I said before, this tends to take the A form, so it's slightly more compressed. And the fact that you can form these self-bonded double helix regions allows you to take several different conformations. So here, for example, we have one strand here, and you can see it has AUGCCA. Here we have a sequence that has complementary bases, UAC, then it has a bunch of bases that aren't complementary, and then a few more that are, so GGU. When this binds together, this will leave this little bulge loop, and it'll leave these nucleotides exposed, and this can serve as a binding point for some type of protein that is recognizing this shape. We can have a similar type of structure where we have the bulges or these little loops on both sides. So we have base bonding here, so we have double helix here and double helix here. But the fact that these do not bind over here allows them to rotate more freely and they can stick out. Again, it can serve as a binding point for proteins. We can also utilize this base pairing to create more complex structures. So here we have a kind of a multi-branch T-junction type of thing. We actually see this present in transfer RNAs. Here we have a stem loop, so this is kind of a combination of this junction with one of these little bulge loops. So here we have a strand over here, complementary sequences, more complementary sequences here, and then a little thing that sticks out. And this type of thing, these bulge loops or these stem loops, is actually where you see the anticodon in transfer RNAs. But again, it allows also for binding of different proteins. So when we look at the structure of a transfer RNA, which is shown here, you can see that it actually has a very complex three-dimensional shape. And this is important because as you start looking within the cell, you will find several instances of RNAs forming complex three-dimensional structures that can actually do things, working very similarly to how proteins work. Okay? And this kind of leads us to a proposal that perhaps before there were proteins, you could have RNAs forming complex structures that might act as enzymes or structural features. And there is some evidence for this in the microbial world as well. But even if you look at the ribosome itself, the bulk of the ribosome is actually made up of chunks of RNA folded into an enormously complex shape.